Hi, everybody. Today I've got Ryan Zenhausern. He's in the same lab as Katie and I, and he is going to introduce himself. Ryan?
How's it going, guys? I'm Ryan. I am a fifth-year accelerated master's student at the U of A in this Biosensors lab, and I'm here to talk to you guys today about diagnostics for cell therapy. So some exciting stuff we're working on.
Oh, yes. So excited. Okay, yeah. So we're going to just let you get the ball rolling. And before we start talking about NK cells and all this cool stuff, can you just explain the immune system a little bit to give us a bit of background?
Yes. Okay. So all right, let's see. Where to start?
The
Hold
immune
on.
system regarding how you use it and stuff.
Okay, great. So for those of you who might know, the immune system is very helpful in fighting off infections and diseases and cancers, right? Often this gets dysregulated in certain disease states, and so your body can't fight off what it's supposed to fight off in the right way. And so researchers are out there now developing these new cells that are really smart, right? They have all the information they need to know, I'm going to go to that cell and I'm going to kill that cell. I'm going to kill that cancer, right? And so that is generally called cell therapy. This is really exciting, and it showed a lot of promise in the past. For example, on a lot of blood cancers, they use these CAR T cells, so these chimeric antigen receptor T cells, which just have a modified little bit on their membrane, and they know where to go. Recently, people have been trying to find other immune cells that are going to be super helpful for this kind of therapy, but that don't have the same kind of immune rejection that certain other therapies do. And one of these promising cell types is the natural killer cell, the NK cell, which everybody has in their body, right, already. But they want to engineer these and put them back in to target your cancers. And so we are looking at trying to find a way to quantify these cells to make sure, like you would say, I'm going to take 10 milligrams of Advil. You want to know how much drug of cell you're putting in your body, right? Yeah, and so having a sensor to do that would be really helpful.
Okay, so just to make sure I'm following correctly, you said there's an immune rejection. Is that when the cells that they put into the body doesn't like?
Yeah, and so basically they realize your body is smart enough to know, like, hey, these aren't our normal friendly cells. There's something weird here. And so these engineered cells, they kind of wreak havoc, right? Your body will have some cytokine release, like that there's in COVID, for example, right? Where your body is just kind of sending a lot of stimulatory cues to your immune system, like, hey, the alarm, something's going on. And then you will have like a lot of neurological problems. You'll have kind of fevers, things like that are very painful and very like damaging to your other organs.
Okay, so cell therapy, it's good for you, but there's a big issue that you need to know exactly what cells and how much to put in, like that Advil example you gave.
Exactly.
Okay, I follow that. Oh, I'm so happy you're talking about this. Like, okay, this is cool. So, okay, then types of NK cells?
Yeah, great. Okay, so let's start there. So these NK cells, they're not just kind of like any cell type. It's not just one thing. There's actually a distribution, and this is, what differentiates an eye cell from a blood cell is kind of the receptors and the channels that are on in the cell, right? And so even for these natural killer cells or these NK cells, there's kind of two main subtypes, some that we'll call bright and some that we'll call dim. And that's just relative to their expression of this certain surface marker that's CD56. You don't really need to know that. Just know that there's a bright, has a lot of it, and then a dim that has only a little bit. And so these two subsets, they kind of do complementary things, right? So when you reach an antigen or something that's like, presenting in your cell that you want to get rid of, the bright subpopulation, they release these cytokines, right? And they're stimulating everybody else, saying, hey, they're over here, like, go get them, right? And a lot of other cells will kind of collect around that and attack them. Versus these dim cells, they're the ones that are directly going in and attacking. They're the ones that are eating up all this garbage in your body. And so
they've got different jobs.
Exactly, they've got different jobs with the same kind of ultimate end goal, right? And so when we make these NK cells in your body, right, depending if you have cancer, you might have more of one population over the other. So for example, they call these bright cells, they're just kind of immature dim cells, right? So they're the precursors. So in your body, if you have a lot of the precursor, then that means that your active form of it, the dim cells, are dying more frequently, right? So that means your body has something wrong with it, right? And so by measuring the actual proportion of your bright to dim, you can tell that there's something wrong with you, right, in your disease state. So when it comes to cancer, if we're injecting these cells because we want you to get better, but we can tell that it's not, like, the proportions aren't right, that you're actually having other problems, right? And so we should take a look at you and maybe refine what we're doing to help you out more.
And if you're doing all this to try to create sensors that can diagnose the number of bright cells and the number of dim cells.
Yeah, so we want to ultimately tell you how much drug do you have in your system of cell, right? And then also kind of prognostics, where you're saying, like, how well is this drug working? How do we need to modify the drug regimen, right? So we should add more cells, or we should add more of these kind of cells, right? Yeah.
And this is from blood?
Yeah. And this is all gonna be from blood
Crazy.
samples, right? Yeah. And so, I mean, the sample we actually use in lab is like Buffy coat, which is just like mostly the white blood cells, but it's a
Okay.
blood-like matrix. Yeah. So that's pretty cool.
Okay, that's awesome. Okay, more about the CD56, I guess then. Can you, I think I follow what they are, but when we were talking about this before, you gave the most amazing examples of kind of like army troops and uniforms. I think you should
Okay.
give that
Oh,
analogy.
okay.
It was
Okay.
on
Okay.
point.
Yes. Okay. So that's what I always said is like, so like a lot of people say like immunology is a lot like, fighting the battle against whatever, it's really easy to kind of envision that, right? And so in our example, CD56 is the thing that differentiates like the Navy from the Army, right? So we know if we have the Army, they're all CD56. And then our two subpopulations are the bright and the dim, and it's kind of like their uniforms, right? Like those that are really getting down and dirty and fighting in the trenches they have like a lot of dirt, right? So their uniforms aren't as bright as they used to be. So they're the dim cell subtypes. And then the bright cells are like those commanders that are telling everyone, hey, like over there, So that's an easy way to think about it or remember it.
I live for the
Yeah.
analogies. That's perfect.
Okay, yeah.
Okay, and so then how do you see them? That's
Okay,
get into that.
totally. Okay, yeah. So let's get into like that's what we want to do. We want to tell how much of each population, right? And so unfortunately, it's really hard to just look at a glass slide and be like, yep, there's a lot of cells in there, right? And so we kind of need something to tell us what's there, right? And so our lab uses, I mean, if you might see in other videos, if you watch other videos, you might see, I know, right? Yeah. We use a lot of times, we use these fluorescent nanoparticles, right? And so these are just little balls of like fluorescent dye. And we put antibodies that are specific to our cell on there, right? So in this case, we're looking for these CD56 NK cells. So we have CD56 or anti-CD56 nanoparticles. And so we add our particles to our cells, they kind of surround them and they tag them and say, hey, look, they're all right here. And when we excite those particles, we can see a lot of light. And so then we can tell how many cells we have. So in this case, we want to know if we just lumped our whole sample together, right? We would just see a lot of fluorescence, and we wouldn't be able to tell, what's the proportion between bright and dim. So in this case, our sensor, we need to separate those two populations, right? And so this is kind of something that we thought of, interestingly enough, using our paper-based microfluidic devices, that when you think about it kind of intuitively, things that are big and heavy, if you were to try to flow it down a river, will flow more slowly than if you threw a stick in, right? Very light, very fast, and that will flow further. And it's kind of the same thing with our cells. Those bright cells, not only are they a little bit bigger, they'll have a lot more particles attaching to them, right? They're much more cleaner uniforms. We can really get in there with those particles. So those guys are big and heavy, and they'll stay to a different part of the chip versus those light ones, those dim, will flow much further, right? And so if you were to just put a sample on a piece of paper, those bright cells would stay right where that drop hit, and then those dim cells would flow outwards, right? Because they're getting pushed by this water, by this river of fluid, right? And so we designed a chip that kind of is just a single lane, and it's the same kind of concept, right? So if you take one image from the inlet and then one sequentially until the outlet, you will see kind of a profile of the cells. Oh, we see, 10 bright cells in the first image, and 1 in the second image, right? And so you can tell based on how many particles are in that field of view or how much you're looking at, how many cells there are. And then that way you can tell, oh, we have 30% bright and 70% dim cells.
Amazing.
Okay. Any questions? Because I know that's kind of fast, but
no, that's really good. You should be a professional analogy maker. Flow rate stuff has always confused me a little bit as well, but actually imagining it like a river, that's perfect. Yeah, the
right,
bulky
yeah.
stuff is going to be slower and maybe get caught a little earlier.
Yeah, exactly.
That's so cool.
So.
I'm so happy to have you in here. I think one thing that we skipped over a little bit, this sounds amazing. What are the current ways that people figure out what the percentages are?
Yeah, great. Okay, so the way that is done right now is kind of through this big machine called the flow cytometer, right? Same concept where you would attach an antibody that fluoresces that's specific for that thing you're looking for, right? So in this case, we have a fluorescent antibody that goes to those CD56 NK cells. But in this case, this machine is very big, very expensive, and needs to be performed by professionals in like a lab setting, right? And this machine is really good at kind of separating cells and separating out the exact populations, So this will tell you, it'll take cell by cell, one at a time. Yep, you're dim, you're bright, you're in between, with some kind of accuracy, right? And it'll give you much more comprehensive profiles than what our sensor can do, but at the cost of not being available to a lot of people. So this is not something you could really use in your home or to say, for example, monitor your own health if you are a cancer patient, right? So.
Yeah, that's a great point. A lot of the stuff that we do in our lab is low cost and heading towards that home-based healthcare. We'll
exactly.
See.
Yeah, exactly. Something that at least all the practitioners could use, like your doctor, you could do this at your doctor's office, for example. Yeah.
Right, because it's also, not only is it cheaper, it's also a lot faster, right? So if you just need a quick preliminary result, you can use your thing and then later confirm with flow cytometry.
Absolutely. So
that's cool. Okay, well, to wrap us up, I think I would like to hear your least favorite thing about working on this project, a limitation or just something that's annoying, and then your favorite thing about the field in general, like the potential you see for it.
Okay, well, this project has been very interesting for sure. Definitely a lot of pros and cons. I think it's always interesting to do something that excites you, something that you think like is kind of science fiction, right? It's very fun. So I think with that in mind, the only limitation that's kind of hard is, and I think this is with any technology, right? How things develop. We had a platform that worked really, really well when we're taking images that are very controlled, for example, on like a benchtop microscope. So, typical microscope you think about. And then transitioning that to our like smartphone-based microscope, that's been a whole nother set of challenges, So getting the project from its idealized form to where we want it to be was like significantly more difficult than I had anticipated at first, but I guess that's how it always goes.
Classic research.
Classic research, exactly. But I think at the end of the day, it's super exciting to know that like this field can be so diverse, and like when you think of cell therapy, a lot of people think of like, I don't know, I think just making new cell types and like new engineered cells and like gene networks and things like that are like very, maybe not necessarily within our current expertise in our lab, but it's super exciting that we can bring in so many more sides to it to kind of make this therapy go from the lab to something that might eventually be much more commonplace in the treatment of cancer. And that's something that's obviously very exciting for a lot of people. So I guess we'll see where it goes in the next 5-10 years, but I think it's going to go some amazing places. Yeah.
Oh, Ryan, this has been awesome. This is fantastic.
Thanks for having
I know
me.
that you anytime, again and again. I know you've got a paper in the works, and so I'll probably release this earlier, but once that's released, I'll put it down there so people can read it.
Okay, yeah, please check it out. Make me famous. Yeah.
Awesome. Okay, well, thank you so much for coming on, and I'll talk to you later.
Yes, sounds good. See you.
Do.